the button. It's green. I pushed the button. I'm an experimentalist, so I know how to push the button. Okay, so I want to tell you about some work that we're doing at Princeton, and along with some collaborations I'll tell you about. This is funded through various agencies. It's sort of a strange set of projects. As you'll see, this is weird. Those new batteries are not so new. That's okay. And in particular, it's happening at an NSF Physics Frontier Center that we have at Princeton called the Center for the Physics of Biological Function that I run. Yeah, I'll take that. Oh, this one's even better. It's like the same, but better. Okay, hold on. I tried what I thought were fresh batteries. But, oh, that's amazing. Maybe not so much better. What? Okay, good enough. Magic. So much magic. Okay, very good. So, so I've been at Princeton for 12 years, which means that I've been a useless scientist for 12 years. But these amazing people, thankfully, still do interesting things in the group. I'm going to tell you about a lot of work we've been doing over about 10 years. I've actually been working on this organism for 20 years at this point, I realized recently, which is interesting to have a bug that you've loved for 20 years. And so some of this is a collaboration with a group in Marseille run by Tam Mignot and some postdocs in my group, Fabian and Matthias, and then some other postdocs in my group, Shashi and Mingzai, who are both now faculty. And then we have more recent collaborations with two theory groups, Ned Wingreen at Princeton and Christina Marchetti, who was at Syracuse but now moved to Santa Barbara, and a bunch of other folks. Okay. First off, how many of you would call yourselves biologists? Just excellent. Okay. Now Sid, you did not raise your hand. Okay. But this is good. You're the right kind of biologist. So I'm not going to use a whole bunch of three-letter acronyms in this talk because I know you guys wouldn't like it. Rest assured, I know the three-letter acronyms. So just to tell you, if they were biologists, I would have to say this because they would be angry with me for not using the three-letter acronyms. Okay. If you know what this is, please don't say anything. Some of you probably know what this is. This is a little material of liquid metal, something like liquid mercury, which I used to play with as a kid, but you're not allowed to play with anymore as a kid. And this thing, it's obviously attracted to itself, so it's sort of agglomerating. And then it does some really weird stuff. It clearly really doesn't like the surface either because it's going to start to de-wet off of the surface and form more of a three-dimensional droplet. And then the droplet gets more structure and forms this humanoid-looking thing, and finally solidifies and crystallizes into the Terminator, who's going to come and kill us all. Okay, and end the world as we know it. So this kind of seems ridiculous, right? There are no materials that we know how to make or we know of that will do all this, except this is essentially how all structure in biology is made, including you. Okay? It is made by autonomous, self-assembling, moving molecular scale machines that build fully viable animals, plants, bacteria, mushrooms, my son a long time ago, dolphins, whatever. Okay? We do not know how to make these things. Not only that, I would argue we don't even really understand the conceptual physics behind these kinds of active systems. Those are the kinds of things that drive a lot of the research that happens in my group. So let me tell you a little bit about some systems that we do understand that don't quite look like the Terminator. One of them is you. So this is a fruit fly. You guys are a lot like fruit flies. Physicists don't like this, but of course they like spherical cows. So I don't know how to make everyone happy. But this is a fruit fly embryo. It starts out as a bag of cells, and you could see early on there was sort of a line of cells, which is now migrated over here, that was defined right about there. And that's where the neck is going to be, and so this is going to be the head. This thing is rounding up to form a tube. You all know you are topologically a donut. Same thing with the fly. And so all of these cells have to know where to go, which direction to move, whether to divide or not, to form this kind of spatial structure. And the way they do that is by reading a map. What happens is the fruit fly, and you cheated when you were embryos, because your mother laid down a source of chemicals here that diffused and degraded forming a characteristic reaction diffusion length scale and direction that the cells could read out. And there are similar gradients that define the other directions, particularly if you include the chirality of one particular molecule. So the mother lays down a coordinate system, a three-dimensional coordinate system, that the cells in the embryo then read and make decisions based on. And that's how they know where they are and where they should go, and what kind of cell they stay. If I'm here, I should be a neck cell, or I should go over there and become a whatever cell. Okay? So, that's also self-assembling sort of nanometer microscale materials, but of course it's cheating because we've imposed some sort of coordinate system from the ground up. Something slightly more autonomous. These are single-celled amoeba that crawl around a surface called Dictyostelium discoideum, and they are single-celled and autonomous. There is no mother that tells them what to do, and when they starve, they aggregate into large structures that end up looking like this. Okay, and they have to figure out where to go, how to do that with their neighbors. And they don't have a global map that's generated by some mother. What they do is generate their own map by all sensing the concentration of one particular molecule, and then with a time delay and an amplification, making more of that molecule. And when you put the collective together and all those little rules locally together at the cells, you generate in space a concentration pattern that looks like spiral waves of this chemical. So as a collective, these cells then generate a map. And then they can individually read the map. Okay? So these don't look quite like the Terminator. They approach the Terminator, something like that, but not quite like that. So this is the star of our show. This is Myxococcus xanthus, my favorite organism, not just cell, organism in the whole world. For 20 years, Myxo and I have been very good friends. And it's a single-celled, wiggly, rod-shaped bacterium that crawls along surfaces. But this cell lives in groups, and it must live in groups. The reason it lives in groups is because it eats other bacteria. So Will sometimes, as you know, studies E. coli. Myxo eats E. coli, so Myxo wins. This is a colony of E. coli, and you may also know that bacteria move around by swimming with these helical propellers. That's essentially false to first order. Most bacteria don't move at all, actually. Some of them swim like E. coli, but I've put them on a solid surface. There's not any liquid around, and so the E. coli are stuck. Now this is a colony of Myxococcus, and they would like to go eat the E. coli, and so they are going to crawl and swarm and devour and kill that E. coli by excreting antibiotics. The antibiotics essentially make the E. coli blow up, and then the Myxo absorb just whatever kind of comes out of it. Okay, so that's one version of eating. And a single E. coli, or a single Myxococcus, this guy, generates a very small concentration of antibiotic and is not very effective at killing anything. But 100 million Myxococcus can kill a lot. So they need to hunt with their friends in order to live. And you'll see these guys will swarm out here. They will cover the E. coli. They're going to form a bunch of traveling waves that we will talk about at the end of today's talk if we get there. And they are actively killing all of the E. coli here. So here are the waves that you find. Now, these E. coli were not very lucky. These Myxococcus were not very lucky. These Myxococcus did not make it to the E. coli, and so they didn't get to eat, and so they're hungry. They're starving. And what these did in large groups of between 10,000 and 1 million, those cells formed round droplets that de-wet off the surface. And inside of these droplets, the cells do what we call sporulate. They essentially become little round protective things that can survive without food for very long times. Okay? Now, they could be starving. In fact, some of these cells out here will eventually form spores all by themselves. They could just form a spore. But the problem is when they decided there might be some food and they woke up or germinated from that spore, they wouldn't be near any of their friends. They would not be able to effectively hunt, and they would die. So they have to form spores together, and that's what they do in this droplet. The droplet is both protective and a way of making sure that they all stay together so that when they do germinate, they have a large number of their friends. Okay? And so we're going to talk a lot about the motion and the droplet formation and things like that. Is everyone happy with the quote-unquote biology of Myxo? Okay, very good. So we spent a long time studying how Myxo move. There's a bunch of papers you can look up about this. They have two different motion systems. One is they polymerize a grappling hook polymer that's extruded, a single polymer from the edge of the surface. It binds to the outside, and then they take it apart at the cell surface, and they hold on to the end, and so it pulls, literally a grappling hook. Okay? And they can use this to pull themselves ahead and to bind to other neighbors and things like that. And they also have a group of little feet, essentially, or tank treads that run from the head to the tail of the cell. Those tank treads can bind to the substrate, and they push it along. Okay, and they use both of these things. They can control the direction of both of these things, and they can control the speed of both of these things. Okay, cool. Amazing, right? I mean, we don't know how to build these things. It's pretty slick. Okay. They also can reverse their direction. Literally, they take apart the motors at one end of the cell and put them on the other end of the cell, and then the cell goes the other way. And they do this sort of periodically. It's not Gaussian. It's sort of exponential sometimes. Sometimes it has a little peak at intermediate times. But they go backwards and forwards like this. In fact, how many of you you talked about chemotaxis today? Did you talk about the key system at all? Okay. Anyway, I told some of you guys today about this amplifier that can change the offset. Yeah, when we're talking about chemical sensing in E. coli. Well, that's an amplifier. And what happens if you overdrive an amplifier? Anyone can answer this question. It oscillates. So this is a chemical amplifier. It's based on methylation. All of these things that say FRZ are actually the same amplifier in E. coli's chemical sensing amplifier, except it gets overdriven so it oscillates, which is kind of cool. And this you can actually see in experiments, which is sort of a neat thing. Okay. So in addition to moving and going back and forth, they interact with each other, and they have two modes of interaction. In one mode of interaction, they have essentially nematic alignment because they bump into each other and they're flexible and they end up pointing in the same direction. Okay, so they have a nematic alignment term. As far as we tell, there is no polar interaction, i.e. one's going like this, do the same things as one going like that. So any asymmetry is actually very small. And when they move, they dig out. Is this going to start? Here we go. They dig out pieces of the substrate. So in red here, so this is a height image. We can actually measure the height of everything going on. And as the cells move around, you can see they leave behind a divot or a trench behind them, and they follow each other's trenches. They kind of fall into the trenches. And so they both repeat their own paths, and new cells will follow the path of old cells. And this is an interaction that will be important for one of the phenomena that we study. When they're moving on a substrate, this is important. When they're moving on each other, this obviously isn't very important. Magic button, do something interesting. Okay. I think I just said that. Okay, not so important. Okay. Okay. So the cells move. The cells have a nematic alignment. And just like birds or fish, which have nematic alignment because they want to, these ones have an alignment because they try, they just run into each other. You should be able to get collective active states like flocks for the same reason that you get bird flocks. Okay, and a lot is known about sort of the physics or the statistics of flocking phenomena. This started in earnest sort of in the mid-90s with simulations. You could just make simple simulations of little vectorial things that want to partially align with each other, and you'll get things that look like bird flocks. And that's kind of where it started, and then the computer animation industry added a lot, apparently. What you should think of is this is like an Ising model, where locally you have an interaction that's trying to align these vectors with each other, a polar interaction, except that now I move along the direction that I'm pointing. Okay? And it's this movement that allows me to do things like break symmetries that are not allowed in a thermal system. Okay? And this is going to keep coming up today and today. What happens when you burn energy? You're allowed to do things that you can't do in conventional thermal statistical mechanics, and you can make active states that break symmetries and do all sorts of cool stuff. Okay? Now, one characteristic of some of these active flocking situations, you can get lots of different phases. You know, you can get a locked solid state where they sit there, and they're all just sort of not really moving. But then you can get a moving solid, where the flock stays locked in sort of relative position and just moves as one cohesive thing like this. You can also get liquid-like states where you have partner exchange, neighbor exchange of position, and you can get moving liquids, which is what a normal flock, if you watched a flock in sky, right? So the moving solid is like the V, you know, the geese that make the big V. They don't change very much. And then a normal bird flock that's doing all this cool stuff out in the wild will be more like a moving liquid. And in all of these systems, if you have a collective state that is in 2 or more dimensions, you have to have what have been called giant number fluctuations. These are anomalous size fluctuations in the number of elements in the groups. And this comes from the fact that you're out of equilibrium and you can sort of go through all sorts of math. And this must be true. So we took Myxo and we put them at sort of low density, and they move around. They have these nematic interactions, and they form flocks. And you might say, well, wait, why do they form flocks if they have nematic interactions and not polar interactions? And this is because polar particles that move with purely nematic interaction form polar states for the following reasons. If I have all my cells lined up like this, they move together more or less, and they have a nice flock. If I have 2 flocks like this, they look like good flocks. At this point, there's no more order, but then I wait a little bit, and now there are 2 flocks again, because they move along that direction. So you get effectively a polar state without polar interactions, which I think is kind of a cool statement. And in fact, you can look at the size fluctuations of these groups, and indeed you get the kinds of anomalous fluctuations that you expect in 2 dimensions, because now we're talking about 2-dimensional flux. If you allow the cells to go backwards and forwards, what you find is they don't form flocks anymore, even though the interactions are all the same. So here is a faster movie of these kinds of flocks. They're sort of unidirectional. Now you can see the fluctuations much more. They like join and split up and have all sorts of interesting dynamics, even though locally they're all sort of moving together in these polar states. And then if I make the cells reverse, so what I did here was I took Myxo and I broke the clock, the oscillator. So now they can't oscillate anymore. And then now this is the sort of what we would call wild-type cells, the normal cells that can't oscillate back and forth. And when you do this, instead of getting these flocks that move everywhere, you get these static, what we called streams. Another group called them rivers, where essentially the cell density is local in space, and the cells are moving in 1 dimension. If you look at where those cells, I forgot to reverse this. These are the flocking cells, and these are the streaming cells. If you look at where the cells are in space, they're essentially always in the same place. Not a same cell, it moves all around, but it moves in the streams, and it doesn't go outside the stream, basically. So what the cells have done here is they've seeded local density fluctuations that are long-lived, which they're going to then use to form the droplets. Okay, and flocks don't let you do this, because they're always just sort of moving everywhere. And you can even show that in this, because it's pseudo one-dimensional, you don't get anomalous size fluctuations anymore. You don't get the giant number fluctuations, because it's in 1 dimension. And those are only required in 2+ dimensions, which I think is kind of a neat result. So we spent a while thinking about why these streams formed. We did some simulations where I thought, well, maybe they just stick to each other along the sides or on the heads or the tails, and that just makes them clump up. And so that doesn't do it. But if you have this sort of a trench following that we talked about, then they very quickly lock, the reversing cells quickly lock into these streaming states that are really, really long-lived. Okay? And then these grow over time very slowly. The width of these will grow over time and just sort of grow and grow and grow, but stay at the same place in space. Okay. So let's add some more cells. Okay, so this is now almost a single layer of solid cell. So this is the ground, the substrate underneath them, and then the rest is cells. And you can see there's large amounts of nematic order, right? For those of you of a certain generation, this looks like a liquid crystal, right? Except what's kind of neat in this system is we get to see the molecules. Normal liquid crystals, you don't really get to see where the molecules are simulated. Okay, so here we see them, and now we're going to take this liquid crystal, and we're going to make it an active liquid crystal, because all the cells are moving. And you get all sorts of crazy dynamics. This is one of my favorite movies ever. I watch. Sometimes on the airplane, I just stick this on and I loop it, and you know, people leave the seats around me. Who is this crazy guy? But look at all this, right? Holes open up, holes close, layers of cells form on top of other layers, you know, through this active nematic. So we just starting this summer, we were able to take this kind of data. So I'll tell you about what we're doing. The results I'm going to tell you about right now in the next 10 minutes come from just a few experiments. We now have many more experiments, and they're all running on the cluster. So soon, you know, we'll have more data. But I think all the conclusions will be the same. You guys seem to like this movie. I'm going to leave it up. It's really good, right? It's so cool. All right, so if you know anything about liquid crystals, you know that it's the topological defects that govern a lot of the behavior of these liquid crystals. Defects, disclinations, defects in the nematic structure that tell you what the behavior of the liquid crystal is going to be like. Okay? And so in this kind of two-dimensional, purely liquid crystal like this, there are multiple kinds of defects you can have, and they are quantized by what we call half-integer topological charge. And so you can have a plus 1/2 or a minus 1/2 topological defect. A plus 1/2 looks like a comet tail. So these lines here are meant to be the director field, which is the local orientation of the cells. So if we look here, here you see a plus 1/2. There's cells coming in. These cells are sort of pointed vertically, and around it they're spiraling like this. Okay? So that director field looks like this. If I do an integration from this point back to itself, I have to take the director field and rotate it by half of a full turn. That's where the half comes from. So the charge is this KB here. There's another way I can rotate by a half turn, and that's by rotating in the opposite direction that I'm doing the line integral. So here I'm rotating the line integral this way, and I'm rotating the vector the opposite way. Okay, and I can do that half a turn, and what I get looks like a trifoil. Okay, it's a three-pointed thing, which, let's see, if you look carefully. Tell me if you see one before I do. Here's one right here. Okay, so this is a three-pointed thing right there. So that's a minus 1/2 defect. Okay, you can't have anything in the middle, right? Because like if the director field points like this right here, if I make a full turn, I can be this or another one, but I can't be in the middle. So it has to be at least half integer. You can get full integer defects. We do sometimes see these. They're very rare. In a conventional liquid crystal, the energy it takes to make a defect like this, with a thermal fluctuation, the energy is the topological charge to the 4th power. So they're quite expensive, and you rarely get things that aren't just the 1/4 power fluctuation. So that at finite temperature, you have some equilibrium number of these defects. And it's also true you cannot just make a single defect. In other words, the topological charge of the system, if it was really a closed system, is conserved. So if I have a whole bunch of matchsticks that are all pointed up, and I start futzing with them around, I cannot make just a plus 1/2. I can make a pair, plus 1/2 and a minus 1/2. Around it, everything will be vertical, and I can, if we take this thing and rotate it around and put it over there. So let me get a chalk. Okay, so I have, this is my minus 1/2, right? And then I'm going to make that like this. So this is a minus 1/2 and a plus 1/2. And I can take a fully vertical thing and stretch this little comet here, and I get a minus 1/2 and a plus 1/2. Right, to happen, but the total charge is not conserved because minus 1/2 plus 1/2 is 0. Yes? Okay, good. And I could do the same thing with this, slightly more complicated. All right. So we're going to take images that look like this. Okay, so this is another movie, also really cool. We know not only the director field of the cells, but we can measure the height. This is a reflected light technique using a confocal imaging that allows us to measure the actual height of the substance. And so then what you can get is you can tell when the different layers form and where the holes are and that sort of thing. So there's going to be a second layer that forms up here in a little bit. We'll start this one later. It takes too long. There it is. Okay, so another layer forms over there, something like that. Okay. Then you can look at the direction of the cells in the image and calculate the director field. So here, the color tells you which direction the cells are pointing. So blue is horizontal, yellow or red is sort of vertical, and then green and red go between them. Okay. So defects are going to be points where you have a pinwheel of color around a single point. And depending on whether that defect goes, red, yellow, blue, yellow, red, purple, or blue, purple, red, yellow, tells you whether it's a plus 1/2 or a minus 1/2 defect. Okay, so then now our beautiful movie looks like this. And what you're going to be able to see here now are the pairs of topological defects that form. These little pinwheels will pop up, do things, they interact with each other. Now, I said before the topological charge was conserved in a closed system. This is not a closed system. One is there's lots of things outside that we don't see. But there's also these boundaries, and the boundaries can absorb topological charge of half-integer multiples. And so you will often see that the topological charge of a whole is multiple integers. So that last one was a -1. It had 2 -1/2 sort of the edge. So you did the whole integral around the whole thing, -1. These black lines denote where the second layer formed. And maybe there's a third layer that forms later, something like that. Okay? And one thing you'll notice, which is quite amazing, is that the nematic order parameter is retained across the boundary. Okay? If you look closely at the data I'm not showing you here, what you see is the cells are slightly interdigitated, meaning that not only do they have a nematic alignment interaction in 2D, they have a nematic alignment interaction in 3D. And so what we think is in the large droplets, we should be able to see 3-dimensional defects, which have all sorts of cool things about them. We haven't been able to see them yet, but that's the prediction. So this is another movie you can just watch. They're little defect pairs that do all sorts of crazy things and interact with each other. Okay. We'll go. Hello. Apparently it likes the movie so much it won't stop. Okay. So here is a topological defect pair. We have a -1/2 that goes blue, green, yellow, red, purple, and a +1/2 that goes blue, purple, yellow, green. Like this, you can see this one has the 3-fold symmetry. This one has a comet tail pointed in that direction, something like that. So this is essentially what we drew right here, rotated by 45 degrees. Ooh, didn't like that. Oh, that's weird. Huh, magical. Cool. Let's try this again. Okay, good. So in the next images, I'm going to show a blue dot for the position of a -1/2 defect. I don't actually have the 3 lines on the movie I'll show you next, but we do know sort of what the orientation of the defect is. Oops. And I'll show a red dot and a comet tail that describe the comet tail of the +1/2 defects. So now we can watch these things form. So when we start the movie, there are 1. So now it's easier to see. That's a -1/2. That's a -1/2, -1/2, -1/2. And there's 1 +1/2 coming out of there. And we can watch these. You'll see pairs form. So there's pair formation, another pair form, another pair form. You'll see them get absorbed by the boundaries. All sorts of interesting dynamics there. Cool. Okay. Wow, this doesn't like this thing. Okay. You can look at how they move. Because the cells move along the polar field, they're polar particles, not the director field. What you have at the -1/2 defects is essentially diffusive motion. So those -1 defects don't have any directionality to them. They don't have any specific propulsive direction, and so they rotate and translate diffusively in space. The +1/2 defects, though, because they have the comet tail, anyone not pointed towards the end of the comet tail quickly leaves the comet. So the comet is basically directed at the other cells, and so it moves in one direction, toward the way you think the comet should move. And so those move, at least on short time scales, in a more ballistic fashion if you look at, for example, the mean squared displacement. You can then look at the interaction between defects, and just as you see in a passive liquid crystal system, you see that opposite charge defects have an attractive interaction that makes them want to come together, and same charge, so plus, plus, or minus, minus, have repulsive interaction between them, just like in electrostatics or something like that. And so they interact with each other, they move around. And now what's governing the formation and the interactions and the motion in these potentials here, these aren't real potentials because they're not jiggling around with thermal energy, they're moving around with motion energy, right? With motion, directed motion. And so, you could think of this like a potential if you wanted, but it's not. It's not a thermal activation barrier. Okay. So one of the really neat things we see with the defects is that the defects are the places where the holes in the layers form. So at a +1/2 defect, I have this comet of cells hitting these other cells here. There's high stress generated there, and they're more likely to form a second layer as the comet cells climb up onto the other cells. And that's exactly what we see here are 2 +1/2 defects that then end up making 2 second layers. Okay, and we see that essentially all the layers, second layers, are formed by +1/2. Not all the +1/2s form layers because sometimes they annihilate somewhere with another defect or on the edge, but they form second layers. The -1/2 defects, because there's essentially no stress pointed at the middle, are sort of low pressure states, if you think of the active stuff as making pressure. And so that's where you see holes develop. So this -1/2 defect is going to open up into a hole that still has the -1/2 charge, just as big. Okay? And in fact, when holes close up, they almost always have either -1/2 or -1, because there were sort of two -1/2 that join together and then have to resolve. Okay. How are we on the liquid crystal stuff? Was that fun? Okay, I'm going to leave liquid crystals in a minute. Yeah, we're happy? Yes? No. Like around the -1/2? No. We can talk about circulation later, but not really on this scale. They rotationally diffuse. You can track the orientation and they just whatever. I mean, these also rotationally diffuse with some persistence, but they sort of sneak around. Okay, more questions? Good. This thing's amazing. It's so angry. Okay, good. So I want to now focus on these fruiting body droplets, which are at a totally different scale than what I've been showing you. Right? We've been talking about flocks of tens of cells or these liquid crystals of a few thousand cells. Now we're going to talk about droplets that have millions of cells. Okay? And the field of view is going to be, I don't know, 100 million cells or a billion cells. Okay? So we're going to go into the coarse grain limit, we might say here, and ask how we can understand this system. So here's a three-dimensional view using our three-dimensional microscope of fruiting body formation, and you can see you get little flock-like groups, and then they join each other, and they start to grow, and they start to get many cells high, and these things will become eventually droplets that look like this. Who asked to see the three-dimensional picture earlier? Who was it? Someone in class. Yeah, there it is. Good. Okay. I have more I could show you, but now the moment has come. Okay. So you can take a three-dimensional picture of these droplets, and they look like droplets, right? They have sort of a pretty constant contact angle, actually. If you look across the population, they've come up off the surface. You can look at the temporal evolution of their height, and they really do just sort of grow and grow and grow and grow, and they, in the end, can be macroscopic. So if you come to my lab at Princeton, I will just go downstairs to these random boxes. I will open them up and show them to you, and you will be able to see these little dots, and that is the fruiting body. In fact, Myxococcus lived in essentially every pile of dirt on the planet. Many things that you have seen in your life that you thought were little tiny fungi are actually myxobacteria of different kinds that form these little mushroom kind of looking droplet things. If the way you discover myxo or isolate myxo is you take dirt, random dirt, maybe not -50 dirt, but something a little better, and you stick it on a plate that has lots of antibiotics and antifungals, and the myxo will crawl out and make fruiting bodies. And you're like, yeah, that's cool. Okay. So we know something. Okay. I don't know what I just said okay to. But, other things de-wet that have nothing to do with little crawling bacteria, right? So if I put water on your raincoat or your Rain-X-covered windshield or a waxy leaf or honey on buttered bread, it doesn't like to spread out and wet the surface. It wants to de-wet off of the surface and form little droplets. Same thing with all sorts of other interactions. And this has to do with the energetics of these molecules binding to each other versus binding to the surface versus the energy you pay to have an air interface, for example. And so you have surface tension and wetting energies. And the combination of these things tell you whether you want to de-wet or not de-wet. And conventionally, you'll make a phase diagram like this. Okay, students, the next two slides are for the people I just talked to earlier. Does this diagram have any meaning to you? Raise your hand if you were in the class an hour ago and this thing has a meaning to you. I didn't think so. Okay. This is called the phase diagram. And in a conventional system, let's talk about a system that I'm going to boil. A bunch of water, I stick it on the stove, something like that. Okay. I have temperature that I can control, and then I have the density per molecule. If you think about water, when it's in the liquid state, it's more dense. And when it's in the gas vapor state, it's less dense. Okay? And you could imagine I could put the system in the middle if I wanted. I could try to make the average distance per water molecule something intermediate. We'll talk about how that happens in a minute. And depending on what the temperature and the density is in these systems, I can get different phases. If the temperature is high and the density is low, I will be in a gas phase. If the temperature, if the density is high, I will be in a liquid phase. And if I'm in the middle inside of this gray line, what I'm going to have are regions of space that have liquid in it and regions of space that have gas in it. Okay? And let's ask why that happens. If this is the free energy diagram where I have free energy here, the energy it costs me to make the system, and the density here, or the volume per molecule, however you would like to say it, I can have one state here, let's call this the liquid state, and another state here, let's call this the gas state. And in the middle, the energy is high, meaning it's unfavorable. Right? So if I take the system and I try to make the volume per molecule this value, for every molecule, I literally glue the water molecules in some grid that has this spacing, right? The cube of this number as its linear spacing. That will be this energy. But now for the same total volume, right, which is that little volume times the number of molecules, I can take some regions and make them this volume, and other regions and make them this volume. And there'll be some linear combination of this that then I pay the linear combination energy as well. Some of them are in this state, so I add up that energy. Some of them are in this state, I add that energy. And that will drop me to this point on the line that connects these two here. And that's a lower free energy, and so that's the state that I'm going to want to be, that the system's going to want to be. Okay, and so these are the states that are inside here. In fact, this gray line here are these two minimal endpoints there, okay, the minima point. Now, the dynamics of this process depend on where I am in this system, and it depends on whether I'm before or past the inflection point of this line, right? Take the second derivative, see if it's positive or negative, concave up or down. If I'm outside here and I have a small density fluctuation, so I take this little patch, and now I compress this bit into the liquid state, and I expand this bit into the gas state, that admixture of slightly more liquidy and slightly more gas is actually higher energy, right? It's the sum of this thing and this thing, the average, and so it's that energy which is higher. So that is not energetically favorable until I get a very large fluctuation. So I have lots of liquid and lots of gas somewhere, and that will then be energetically favorable. So it takes me a long time to seed a large nucleus of liquid, and that nucleus will then grow. So this kind of kinetics is called nucleation and growth. If, on the other hand, I'm past the spinodal points, I'm on the concave down part of the free energy diagram, then every little fluctuation I get, this admixture is automatically lower energy, and that's automatically stable. So on the microscopic scale, all fluctuations in density are stable, and I will just grow everywhere, and that's called spinodal decomposition. And so on a phase diagram, you would draw this other line, which is the point of the inflection point. Okay. So what does this have to do with Myxo? So if you took a conventional water on a waxy substance, instead of density, you would have the film height, or the volume, total volume of the liquid at different temperatures, and that would tell you whether you're fully wetted or whether you make a droplet, okay, or you're in some admixture state. So we're going to take Myxococcus at a sort of low density, and if you really look carefully and have really good eyes, you'll see tiny little gray flocks running around, but they don't do anything more interesting than that. Too low of a density. This would be more of the gas in the gas-liquid state, okay, or a fully wetted system. Aha, disconnect. Okay. Now I'm going to put it at a higher density or a higher volume on the surface, and what I'm going to do is nucleate fruiting body droplets at random positions in space at random times that then grow, okay, as nucleation and growth. And in fact, what you'll see is there really looks like a surface tension where little droplets will dissolve, those cells will go join the bigger droplets, this whole thing will coarsen for a long time. It keeps coarsening until the cells sporulate, then they stop moving. That's like making the temperature 0. They decide at some point to do that, but we're going to look before that time. Okay, so this is nucleation and growth, and now all I do is increase the number of cells, and I do this, and the minute I start watching, there are fluctuations everywhere and everything duets into the spinodal decomposition regime. Okay, so this looks exactly like what you would see in a passive system, except it's not temperature that's driving any of this, right? It's the motion of the cells. Trust me, if you poison the motors, everything stops. Okay. You can measure the length scale that comes out of either regime by taking Fourier transforms of images, and the length scales grow as a power law, which happens in many conventional systems, essentially all conventional systems. And we have decent ways of predicting these exponents. I won't go into too many of those things. We have some publications on those sort of exponents. I'm going to skip this because we're running. When should I finish? 5? Yeah, okay. So I'm going to skip. You can simulate some things, but that's not so exciting. Okay. So what about temperature? If temperature is not the right thing to put on your phase diagram, like we know the x-axis of the phase diagram, it's like volume, right, or thickness of the film, the number of cells we put down. You know, it's a finite volume, so whatever you want. What we don't know is what to do for temperature. And if you think about what the temperature does, it describes the magnitude of the fluctuations of the motions of particles, you know, in this room, for example. And so what we want is something that describes the magnitude of the fluctuations of the motion of the cells. Not thermal fluctuation, but thermal fluctuation just from their motility, fluctuation from their motility. And so the right parameter to do that is called the Péclet number. The Péclet number describes, in a traditional fluid system, the ratio between advective transport and diffusive motion. And here what we're going to say is the cells are liking to move at a constant speed v in one particular happy direction, but that direction is randomized by a rotational diffusion coefficient. Like basically if they try to go in a straight line, they're slightly drunk and they don't totally go in a straight line. And so there's some inherent diffusion coefficient with their orientation vector. But then there's also this reversal frequency. And you can show that what that does to the fluctuations is you take twice the frequency at which it likes to go backwards and forwards, and you add that to this, and this tells you the magnitude of fluctuations, this rotational Péclet number. Okay? So what's really cool in Myxo can control velocity. Each cell can decide, I want to go faster, I want to go slower. And Myxo can control this reversal frequency, meaning the Myxococcus themselves can control the Péclet number, the effective temperature of a single molecule, which is a weird thing to think about in a conventional system. But they can control those parameters as individuals. What we can do as experimentalists is we can control those using drugs. So we make specific mutations that break the cellular control, and then we add small molecules that allow us to quantitatively dial in either the reversal frequency or the velocity. And so now we can sort of ask what happens when we put these systems in different places. And what you do is you can try different densities and different Péclet numbers that we set by adding in these molecules with these different mutants. And then red squares are experiments where the system forms droplets and phase separates. And in the black circles are where it doesn't. And importantly, you can make it phase separate by increasing density or by dropping this temperature like Péclet number. Okay, and we can do that either by changing the speed or the reversal frequency. Okay, so now you ask, well, when did they form these droplets? The cells formed the droplets when they were starving, right? Because they wanted to sporulate inside the droplets. So what happens when the cells are starving? When the cells are starving, we look at the blue dots here. The speed goes up, the reversal frequency goes down, and this inverse rotational Péclet number goes from this value and drops by almost an order of magnitude, factor of 6 or something. Okay? What that does is it takes the population from outside the phase separation boundary into the phase separation boundary, and now the whole thing phase separates. So let me try to explain what this means. What if I told all of you, the next time you are really hungry, run really fast as straight as you can? That's what Myxo is doing. You don't have to talk to any of your neighbors. You just ask yourself, am I hungry? It's been a while since I ate. You hungry? You hungry? Yeah. You say, crap, I'm hungry. Run really fast. And if you all do that, the whole population de-wets into a big three-dimensional droplet. You don't have to make a map. You don't have to talk to your neighbors. You don't have to make a collective decision. You don't have to do anything but run into people and let the physics take care of it. And that's what Myxo does, which is really, really cool, I think. So not only does Myxo know active matter physics, they know what parameter is the one that moves you around in the phase diagram. And they develop control via evolution to change that parameter. You know, and it's not like you would think, oh, I'm starving, I should slow down, I'm running out of energy. And they do the opposite. They go really fast and then eventually sporulate, which also takes energy, right? So I think that's pretty neat stuff. Okay, I'm basically going to end there. We're working on these waves. The waves are amazing, so I'm not going to tell you about them. I'm just going to put up a movie that shows you. They're surface waves, so there's a deep layer of fluid of cells, and on top of that are surface tension-driven surface waves. a single wavelength that pops out, so there's an instability that you get from the active pressure, the fact that the cells are moving, and the interactions between the cells, which I think is quite cool. So we have some results on that coming out. And then I will end by saying that the cell we talked about today, my favorite cell, is the one that made these things, which are what we see in the lab and what a lot you see out in nature. All of these are other fruiting bodies formed by other species of myxobacteria that are much more complicated, but presumably formed by similarly simple mechanisms, potentially slightly more complicated, but I don't think insanely more complicated. And so I think those are pretty cool. I'd be interested in studying them if we could make them behave in the lab, which we're trying to do. But, okay, and it's exactly 5 o'clock.